Hello guys, I am Shubham from ShubhVed Tutorials. This video is about conjugation. So before we start, subscribe to our channel and press the bell icon to get notified whenever video is out. So let's get started with the video. So first of all, conjugation is a type of gene transfer in bacteria. Okay, this comes under microbial genetics. There are three types. Let's see. Microbial genetics. There are conjugation, transformation, and transduction. So in this video, we are going to discuss about conjugation. So basically, what is conjugation? It is a transfer of genetic material through direct contact. Okay, through direct contact, and this is unidirectional, right? Unidirectional, one-way transfer. Okay, so this is just a introduction, right? Definition: unidirectional transfer of genetic material through direct contact in bacteria, right? So how this was discovered, how this was came to known, right? So there were experiments performed. So this is a experiment performed by Lederberg and Tatum. This is strain A and strain B. Okay, we can see this two types of strain. This strain is met bio is absent, nothing, minus sign is there. Minus means signs is not included, and here there are plus signs. Okay, threonine, leucine, and thymine, right? So met bio, there are no signs. Means what? It can produce threonine, leucine, and thymine, but it cannot produce methionine and biotin. Why so? So we have to provide the nutrients, then only it will produce this. It cannot produce it by its own. In the second thing, it is vice versa. It can produce methionine and biotin, but it cannot produce threonine, leucine, and thymine. So these are the strains, right? We will just highlight it. Okay. So when we take these colonies and we plate it, see, wash and plate cells on minimal medium, then we will see that no colonies are formed. No colonies are formed. Auxotropic cell. Auxotropic cell means mutant cells. These are not growing, right? Because we have not provided methionine and biotin in the medium. As well as B strain also, we have washed and plate on minimal medium, no colonies. Here too, no colonies, right? But when we mix both the, okay, when Lederberg and Tatum mixed the both the strains, then plated on minimal medium, colonies are produced. See, colonies are produced, all produced this. Okay, means there were recombination, there was exchange of genetic material, and there was recombination formed. So this, when the colonies are produced, right, those are known as prototropic. Okay, we can say wild type, and this is auxotropic means mutant, right? So what will happen? I will just sum it up here with experiment. First is met bio, threonine, leucine, and thymine, right? Second, cross, okay, cross between this met plus bio plus, and these are just, we can write minus here. Okay, means it can grow in these colonies and it can grow in this, means it can produce this, not grow, produce, but it cannot produce methionine and biotin. First colony strain A, and second cannot produce threonine, leucine, thymine. Then what happened after this? The colonies are produced because all were plus, like this. Okay, so this is due to recombination. We can see here, after the cells from strain A and strain B were mixed and mixture plated, a few colonies grew on the minimal medium, indicating that they could now make the essential constituents. These colonies are recombinants, right? Recombinants produced by exchange of genetic material between the strains. So there was exchange of genetic material, and this was the first discovery of conjugation, right? Then The next experiment to confirm this was performed by Bernard Davis. Okay, this is known as YouTube experiment, showing that physical contact between the two bacterial strains of the Lederberg and Tatum experiment was needed. So contact is important. How he came to know? Let's see. So this is a tube, right? In this part, there is a filter here. See filter, fine pored filter. Okay, means it is having fine pores. Right? Here is a strain A on the left side, and on the right side strain B. Right? Then what happened? After this, again they did plating. Right? Here is the porous cotton plug, and here is the pressure or suction. Right? After this, they did not found any colonies. No colonies were produced. Right on the plate, because there was no exchange of genetic material. Why? Because contact between those strains is blocked. Okay, the contact is blocked by this filter. Whether it is having fine pores, but the pores are not large enough so the bacteria can pass. Right. So that confirms that it requires direct contact, cell to cell contact. Physical contact is necessary. Right. From this experiment by Bernard Davis. Okay. So conjugation is occurs because of plasmids. Okay. So this is very important. Plasmids. One plasmid is F plus. Right. Uh, what we can say, these are the plasmid. F plus. F plus means this is a factor present. F factor. This is also known as sex factor. Okay. Right. And this produces the sex pili, also known as F pili. Okay. Singular is pillars. Right. So this is produced in the F plus cell. So the conjugation occurs between F plus and F minus. Right. One should be F. The donor should be F plus, and recipient should be F minus. Then only the conjugation takes place. It will not occur in this. Right. No. As well as F minus, no. Conjugation will not occur. So I will just draw here a bacterial cell. This is a bacterial cell. Okay. And this is having its chromosome, and here is the plasmid. Okay. These are double stranded. Do note. Double stranded. Okay. So this is a F plus cell. Then there is a pilus. This is a pilus. Let's take another bacteria. This is another bacteria. Again, having its own chromosome, right? But this is a F minus cell, not having F factor. F factor is lacking. F plus cell have the capacity to form this pilus. This is known as pilus. Singular is pilus, plural is pili. Alright. Then this will make contact with the recipient. Okay. So this is how the contact is made. And then they will form a bridge-like structure. Bridge-like structure is what? Just what we can say. I will draw here again. This is a bridge-like structure. And the transfer will occur from this path. From here to here. You can say tube-like structure also. Then after the completion of this step, this cell will also become a F plus cell, because it will be having, I will draw here, small cell with its chromosome and a plasmid. Right? So this will occur. Thus, it takes place between F plus and F minus cell. So this is the first case, right? So we'll see now exact step what will happen. Transfer of F factor. Okay, conjugation between plus and F minus C. This is not having a F minus cell. F factor. Okay, F factor is absent. So this is a plasmid. As we know, plasmid is a self-replicating molecule, right? Self-replicating genetic material, extra chromosomal. Okay. Then see, this is the one nick form, and then this will get moved inside, right? See. As it is moving, we have to understand that so this is a double-stranded structure, right? This is a double-stranded structure. As one strand is moving, the second strand is forming, right? Means replication is taking place. I will draw every different color. See, replication is happening here also, so that it remains double-stranded. As well as here also, when it is coming here, okay, then also the replication is happening, so that it remains double-stranded. See, like this. Okay, so thus at the last, both the strands are double-stranded in both the bacterial cells, right? So this is a first type, we can say, where the cross is between F plus and F minus. Now, what happens? Genetic materials also get exchanged, right? Bacterial chromosome. Okay, how so? We will see that in the next example. It is known as HFR strain. HFR, high frequency recombination. Okay, what is this? So basically, this is again a bacterial cell, right? Then it is having its own chromosome, bacterial chromosome, which is a double stranded, and a F plasmid. F plasmid with a different color we will draw. Okay, here, right? So what happens? The occurs between these two. This is very rare. Okay, can be seen as a 1 in 10,000 E. coli. Okay, that is cross between this. Then what happens? This gives rise to a new type of strain in which the F factor gets integrated. So like this, the F factor gets integrated into the bacterial chromosome, and this is known as HFR. This is known as HFR, high frequency recombination. So here also the cross occurs between same HFR and F minus. Okay, HFR and F minus. So this is the second type of conjugation. Again, same things will happen. There will be one nick here, as we can see here. See, suppose here nick occurs, right, and this gives rise to this transfer of genetic material, right, and as this gets transferred, again replications occur side by side so that it remains in the double stranded form, right? So this is a HFR, okay, and the third type is a F prime, F prime, okay. So what is this F prime? How this F prime forms now? See, again, I will just draw. This is related to HFR. Do note how so. We will just draw here a single strand, uh, but it is a double strand, right? And here is the integrated effector. Okay, this is the integrated effector. What happens? This has to be removed. Okay, sometimes what happens? It gets removed. It gets removed. It is known as excision. So excision. Excision of F factor produces a loop-like structure. See, okay, 8 excision does not occur accurately in this, and the plasmid comes with a some part of bacterial chromosome. Okay, so this is a factor. It will contain some of the bacterial genes, and like this. This is a bacterial gene. So this is known as F prime. Okay, I mean during excision. Excision means getting out of the chromosome. It takes some of the part of chromosome, bacterial chromosome. So this is known as F prime. Okay, in the picture also it is also written same. F factor integrated into the bacterial chromosome here, adjacent to the lac plus region. See, lac plus region is there. This is a factor and this is a adjacent is the lac plus region. Okay. Now what happens? Excision, apparent excision means not accurate. Okay, excision taking place, but that is not accurate. So what happens? F factor loops out incorrectly. So it includes a piece or pieces of the bacterial chromosome, mm, including the lac plus region. So here in the example, suppose we take so lac plus gene is getting out with the F plasmid, right? F factor. So single crossover generates F lac and F factor, which includes the bacterial lac plus region. See, it is having F lac. So it is named. Also, it is named according to genes it contains. So here there is lac plus. So thus it is named as F prime in bracket. It is written lac. Right. So when this type of again this type of cell will also cross with the next picture we will see F prime and F minus. Right. So again it will get transferred with the F plus factor. So result will be F prime again because it will have the some type. See this gene, this bacteria will be having like gene. Suppose we take example, this will have its own like gene as well as this will get transferred from like will also get transferred. So there will be two like plus genes in this. So this is known as metadiploid. So metadiploid means partially diploid. Partially diploid means when a like this is a F prime like having a like gene. This is having a like gene and this is already having a like gene. So when this like gene will get transferred to this will contain a new bacterial strain. This is a donor, right? And this is a recipient. So when a recipient will contain two like genes, right? Two like genes. So thus it is known as metadiploid. Means when these cases occur with F prime, some genes will be two copies. Two copies of some genes will get transferred, and as well as the bacteria, the recipient will have the its own copy as well as the donor's copy integrated into the F factor. So this thing is known as monodiploid, partially diploid, as well as this type of conjugation is known as abduction. Okay, this conjugation is known as abduction or sexduction. Right. So these are the three types of conjugation we have to remember. Just to conclude here, we will summarize. First, F plus with F minus, right? And when F plus gets integrated into bacterial chromosome, it forms HFR. Then the second cross is between this HFR into F minus. Third cross is when in the HFR when F factor is getting exist. Okay, is it forms a loop. It takes some part of the bacterial chromosome and comes out. So that is known as F prime. And the third cross is between F prime and F minus. So these are the three crosses very important to know in the conjugation. So that's it for this video. I hope this video is helpful. If you find this video helpful, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.